Hello friends, welcome back to a new video, that is MCQs on cell signaling, and this is the part 2 of this series of videos on cell signaling. So the first question of this video, that is, among the following molecular process and biological effect pairs, identify the mismatch pairs. So here you have to identify the mismatch pair. So the options are option A, that is histone deacetylation is matched with activation of the gene expression. Option B, that is protein phosphorylation is matched with signal transduction. Or option C, DNA methylation is used in sex specific control of gene expression. And option D, proteolytic cleavage is related to activation of signaling by peptide hormones. So which option, which matching is not true? So the right answer is option A, that is histone deacetylation is not related to activation of gene expression. Why? This histone acetylation, first we have to know histone acetylation, then we will talk about histone deacetylation. So histone acetylation removes positive charge from histone. So histone, when we add acetyl group into the histone, they imparts the negative charge, so they removes positive charge from histone, which reduces their affinity for DNA, because DNA is also negatively charged, so which reduces their affinity for DNA. Okay, so this can enhance transcription, because now DNA can can be recoiled, that means they open up, so they are ready for transcription. Okay, so acetylation of lysine residues on histone tail is associated with transcriptional activation. So by this histone acetylation, this activation of gene expression is done by this histone acetylation, but by histone deacetylation can repress transcription. It can also be a key factor in ensuring proper transcription. Okay, so this is the main reason. So let's move on to question number 22. So which one of the following statement is true for mitochondrial disease? And the options are option A, heteroplasmy, option B, mitochondrial gene mutates less often than nuclear gene, option C, mitochondrial conditions only affect muscle and nerve tissues, option D, the risk of passing on a mitochondrial condition to the next generation may be as high as 100%. So which one is correct about the mitochondrial disease? And the right answer is option D, that is the risk of passing on a mitochondrial condition or mitochondrial disease to the next generation may be as high as 100%. Okay, so this is true about mitochondrial disease. So let's move on to question number 23. It is tyrosine kinase receptors are option P, that is should be trimerized to be active, option Q, that is play important role in activating signal transduction cascades, or option R, that is have phosphate removing active sites in their cytosolic tails, or option S, that is can be oncogenes. So which arrangement of these statements is correct one? So option A, that is P and Q statement is correct, option C, P Q R is correct, option B, Q only is correct, or option D, Q and R statement is correct. So which one is correct? So the right answer or correct answer is option D, that is Q and S is the correct one. So Q and S is the correct one about tyrosine kinase receptor. So Q means these tyrosine kinase receptors play important role in activating signal transduction cascade. This is true, and this tyrosine kinase receptor can be oncogenes. Okay, so let's move on to question number 24. That is the aggregating peptides or proteins in the diseased conditions contain extensive, option A, alpha helical segments, option B beta sheet conformation, option C random structures, or option D beta helices. So which one is the right about the aggregation of peptides or proteins in the disease condition? So the right one is option B. In disease condition, the protein mainly present in their beta sheeted conformation. Okay. So let's move on to question number 25. That is, Hayflick's limit refers to which one of the following phenomena? And options are option A DNA repair, option B cell senescence in vitro, option C protein synthesis, option D DNA transport. Sorry, RNA transport. So which one is the right one? Option B is the right one, that is cell senescence in vitro. So Hayflick's limit refers to which one of the following phenomena, that is cell senescence in vitro. How? Here we will see. The Hayflick limit is a concept that explains how human cells age. So it states that a normal human cell can only divide 40 to 60 times before can no longer divide. After that, the cell will break down through programmed cell death or apoptosis. So this Hayflick limit is based on the length of chromosomal telomeres, which shorten with each cell division. Okay, so this is known as Hayflick's limit, which is related to cells age or cell senescence in vitro. Okay, so next question देखेंगे. Next is estrogen and testosterone are steroid hormones and are most likely to bind to option A membrane ion channel, option B enzyme linked membrane receptor, option C G protein linked membrane receptor, or option D cytoplasmic receptors. So the right answer is option D, that is cytoplasmic receptors. So estrogen and testosterone are steroid hormones and are most likely to bind to cytoplasmic receptors. Okay. So next question देखेंगे. Next is, which of the following statements is or are true about juxtacrine signaling? So the statements are, first statement is the ligand and the receptor engage in reciprocal signaling in this juxtacrine signaling, or statement 2, that is both the ligand and the receptor are membrane associated protein, and the statement 3 is the ligand gets proteolytically cleaved after binding to the receptor. So which arrangement of this statement is correct? So the option A is 1 only is correct, option B 2 only is correct, option C 3 only is correct, or option D 1, 2, 3 all the statements are correct. So the right answer is option D, that is all these three statement is correct about juxtacrine signaling. Okay, so what is juxtacrine signaling? This signaling is a type of cell signaling that requires close contact between cells. So it also known as contact dependent signaling. In juxtacrine signaling, a ligand on one cell surface bind to a receptor on another adjacent surface. So the signaling molecule is bound to the cell membrane and can interact with a receptor on the membrane of an adjacent cell. Okay. So next question देखेंगे. Next is, cholera is an infectious disease caused by the bacterium Vibrio cholera. Okay, so how does the cholera toxin uh, dysregulate the G protein coupled receptor signaling in the host cell? So how does this cholera toxin dysregulate the G protein coupled receptor signaling in the host cell? So the options are option A, that is modifies the GS alpha subunit which is unable to hydrolyze bound GTP, resulting in increased CAMP level. Option B modifies the GS alpha subunit which is unable to hydrolyze bound GTP, resulting in decreased CAMP level. Option C Cholera toxin modifies the GI alpha subunit which is unable to hydrolyze bound GTP resulting in increased CAMP level, or option D, that is cholera toxin modifies the GI alpha subunit which is unable to exchange GDP resulting in increased CMP level. So which option is the correct one about the cholera toxin? So option A is the correct one. So here we can see this cholera toxin modifies the GS alpha subunit which is unable to hydrolyze bound GTP, so resulting in increased CAMP level. So when the CAMP level is increased, here we can see the mechanism of cholera toxin. So this is the total cholera toxin which have two subunit, subunit A and subunit B. So subunit B is helps to bind this cholera toxin to the cell surface with the GM1 receptor. Okay, so after binding, then the subunit A will enter into the cell and it will bind with the G alpha subunit of G protein, and this will activate the adenylate cyclase, and then this adenylate cyclase, increased level of adenylate cyclase increases the CAMP level, and the CAMP level affect the membrane channels, and ultimately the nutrients which are present in the cell, they will go outside of the cell. That means all the minerals will go out from the cell, and that condition is known as diarrhea. So in this way cholera toxin can cause diarrhea, okay, in our body. Okay, so let's move on to question number 29, that is which information among the following provides the maximum information to do structure based drug design? And the options are option A, 3D structure of a set of active compounds, option B, 3D structure of the target, option C, crystal structure of target ligand complex, or option D, primary structure of the target. So which one is correct about the structure based drug design? Okay, so the right answer is option C, that is crystal structure of target ligand complex. Okay, so which information of the following provides the maximum information to do structure based drug design? That is crystal structure of target ligand complex. Okay, नेक्स्ट क्वेश्चन देखेंगे. नेक्स्ट इज receptor tyrosine kinase is, statement P is, this receptor tyrosine kinase are 7 transmembrane helix receptor. Statement Q is they are integral membrane enzyme, or statement R is they activate their targets via the G protein cascade, or statement S is they are often activated by ligand induced dimerization. So which combinations of this statement is the correct one? So option A is P and Q is correct, option B R and S is correct, option C P and R is correct, or option D Q and S is correct. So the right answer is option D, that is Q and S is the correct one. So this Q, that is receptor tyrosine kinase, are integral membrane enzymes, that is right, and S, that is they are often activated by ligand induced dimerization. This is also true about receptor tyrosine kinase. So let's move on to question number 31, that is we assay the activity of a particular MAP kinase in two different cell types and find that activity is present in one cell type but not in the other. So possible reason for this differential activity includes, that is statement P is differential MAP kinase gene transcription option, that means different MAP kinase gene transcription in these two different cell, or statement Q is different mRNA processing of the MAP kinase transcript, or statement R is differential or different translation of the MAP kinase transcript, or S statement is differential post-translational modification of the MAP kinase protein. So which combination of statement is correct? Option A, P and Q is the correct. Option B, R and S is correct. Option C, P, Q, R all statement. Three of this statement is correct. Or option D, P, Q, R, S all statements are correct. So the right answer is option D. That is P, Q, R, S. All these statements are correct about this situation, that when we assay activity of a particular MAP kinase in two different cell types, find that activity is present in one cell but not in other. These all are the possible reasons. So here we will see so what is MAP kinase? So this MAP kinase is mitogen activated protein kinase, is a family of enzymes which activated within protein kinase cascades that regulate cell proliferation, differentiation and death. Okay, so this is MAP kinase. So next question देखेंगे. Next is question number 32. So insulin receptors substrates, that is IRS 1 and 2 are phosphorylated by the receptor tyrosine kinase domain. So what would happen if the IRS, that is insulin receptor substrates 1 and 2 were phosphorylated on a serine or threonine residue? So what will the consequence if the IRS 1 and 2 were phosphorylated on a serine or threonine residue? So options are option A, that is the insulin signal would be prematurely terminated leading to insulin resistance. Option B, the RAS pathway would be activated prematurely and the insulin signal amplified. Option C, the PI3 kinase pathway would be activated prematurely and insulin metabolic effects would be amplified. Or option D, GLUT4 transporters would remain fixed on the cell membrane and would not be able to be internalized. So which one is the correct? So the correct one is option A, that is the insulin signal would be prematurely terminated leading to insulin resistance. So if we if we phosphorylate the IRS, that is insulin receptor substrate, on a serine or threonine residue, then the insulin signal would be prematurely terminated leading to insulin resistance. So now we will see what is insulin receptor substrate. So insulin receptor substrate, that is 1 and 2 are proteins that act as an interface between activated receptor and signaling proteins. They are substrates for the insulin receptor and other tyrosine kinases. Okay, so let's move on to question number 33. It is, what is the first event which happens to a growth factor or cytokine receptor after binding its cognate signal molecule? And the options are option A, binding of a signal activates the receptor's cytosolic tyrosine kinase domain. Option B, binding of a signal causes the receptor to pick up a tyrosine kinase from the cytosol. Or option C, that is binding of signal causes receptors to dimerize. Or option D, that is binding of the signal causes tyrosines in the receptor's C-terminal tail to become phosphorylated. So what is right about the growth factor or cytokine receptor after binding its signal molecule? So the right answer is option C. So binding of signal causes receptor to dimerize. This is the right one. Okay, so next question देखेंगे. Next is question number 34. So cyclosporin A is used in the treatment of organ transplant patients because it, option A, inhibit TCR expression, option B, downregulates IL-2 production, option C, induces T cell energy, or option D, downregulates antibody production. So which is correct about cyclosporin A? So the right answer is option B, that is downregulates IL-2 production. So cyclosporin A is used in the treatment of organ transplant patients because it downregulates IL-2 production. Okay, so let's move on to question number 35. So the demyelination of the central nervous system white matter produced produced by the canine distemper virus is an example of option A fat necrosis, option B coagulation necrosis, option C Zenker's necrosis, or option D liquefactive necrosis. So the right answer is option D, that is liquefactive necrosis. So what is demyelination? This is demyelination is a loss of myelin, an insulating material that coats many nerves. So it can result from diseases that damage the myelin sheath or the cells that form it. So this demyelination of the central nervous system, white matter produced by the canine distemper virus is an example of liquefactive necrosis. Okay. So next question is the discoloration of the tissue by iron sulfide after somatic cell death is referred to as option A hypostatic congestion, option B imbibition with hemoglobin, option C imbibition with bile, or option D pseudomelanosis. So the right answer is option D, that is pseudomelanosis. So the discoloration of tissue by iron sulfide after somatic cell death is referred to as pseudomelanosis. So what is pseudomelanosis? Pseudomelanosis is a rare benign condition that causes dark pigment to accumulate in macrophages in the lamina propria of the stomach and duodenum. Okay. So next is question number 37. That is the specific condition that occurs subsequent to the inhalation of carbon is referred to as option A anthracosis, option B pneumoconiosis, option C siderosis, option D acanthosis, and the right answer is option A, that is anthracosis. So the specific condition that occurs subsequent to the inhalation of carbon is referred to as anthracosis. So what is anthracosis? Anthracosis of the lungs is black discoloration of bronchial mucosa that can occult bronchial lumen and is associated with bronchial anthropfibrosis. Okay, anthropfibrosis. So the next question is question number 38, that is ciprofloxacin specifically cleaves option A nuclear DNA, option B apicoplast DNA, option C both nuclear and apicoplast DNA, or option D mitochondrial DNA, and the right one is option B, that is apicoplast DNA. So ciprofloxacin specifically cleaves apicoplast DNA. Okay, so these apicoplast are organelles that contain their own DNA. Apicoplast are found in parasites of the phylum Apicoplexa. These parasites are characterized by an apical complex, which is a collection of structures that allow the parasite to involve host cells. Okay, so let's move on to question number 39. It is a newly emerged variant of influenza virus can be selectively propagated from the mixed population by addition of option A ganciclovir, option B Tamiflu, or option C interferon gamma, or option D neutralizing antibody. So the right answer is option D, that is neutralizing antibody. So a newly emerged variant of influenza virus can be selectively propagated from the mixed population by addition of neutralizing antibody. And what is neutralizing antibody? A neutralizing antibody is an antibody that is responsible for defending cells from pathogens, which are organisms that cause disease. So they are produced naturally by the body as part of its immune response, and their production is triggered by both infections and vaccinations against infection. Okay, so the last question of this video, that is which of the following cells secrete E-selectins? And the options are option A eosinophil, option B endothelial cells, option C microglial cells, or option D epithelial cells. And the right one is option B, that is endothelial cell. So selectin is secreted by endothelial cell. So this E-selectin is a cell adhesion molecule that is expressed on endothelial cells after activation by inflammatory cytokines, and these cytokines include IL-1, TNF alpha and lipopolysaccharide or LPS. Okay, so these are all 20 questions of this video, part 2 video on cell signaling. Okay, so thank you for watching this video.